Animal biotechnology. Here are the following objectives that should be met after the video presentation. Understand the concept and history of animal biotechnology as well as the applications and technology involved. Before dwelling to the main topic, let us first define biotechnology. Biotechnology is the area of biology that offers new perspective in improving human and animal health, welfare, and increase the productivity of livestock. It improves the quality of the food we intake, strengthens the ability to treat, detect, and prevent diseases, and enhance the impact of animals on the environment. What is animal biotechnology? Animal biotechnology is the use of engineering and sciences to create modified living organisms to improve animals and develop something for agricultural, industrial, and pharmaceutical applications. Animals has a great contribution in the evolution of biotechnology as well as increasingly benefiting in the field. The advantages of the applications of animals in biotechnology is divided into three primary areas. These are improve human health and welfare, enhancements to animal products, and environmental and conservation benefits. Meanwhile, the scope of animal biotechnology includes all animals such as poultry, livestock, insects, fish, and even the companion and laboratory animals. Who are the beneficiary of this advancement? With the help of the scientists, researchers, and doctors, animal biotechnology has been practiced since the beginning of the domestication of animals. Several tools used in animal breeding, nutrition, and genetics have played an important role in the propagation, selection, and management of advantageous and economically characteristics in livestock. Researchers developed the application of animal biotechnology, which have led to the emergence of three scientific animal biotechnology sectors, which are animal genomics, animal cloning, and genetic engineering of animals. These sectors will be discussed further subsequently.
Next is the technology involved in animal biotechnology. First is transgenics. A transgenic animal is one that carries a foreign gene that has been intentionally and deliberately inserted into its genome. The foreign gene was constructed using the recombinant DNA method. Methods in producing transgenic animal include transforming embryonic stem cells growing in the tissue culture with desired DNA and injecting the modified gene into the pronucleus of the fertilized egg. To better understand the two methods in transgenics, I will discuss it thoroughly. First is the transforming embryonic stem cells growing in the tissue culture with desired DNA. This method involves prior insertion of desired DNA sequence by homologous recombination into an in vitro culture of embryonic stem cells. Stem cells are undifferentiated cells that have the potential to differentiate into any type of cell, somatic and germ cells, and therefore to give rise to a complete organism. These cells are then incorporated into an embryo of the mitosis stage of the development. The result is a chimeric animal. ES-cell-mediated gene transfer is the method of choice for a gene inactivation, the so-called knockout method. This technique is of particular importance for the study of the genetic control of developmental processes. This technique also works particularly well in mice. It has the advantage of allowing precise targeting of the defined mutation in a gene via homologous recombination. The next method is injecting the modified gene into the pronucleus of the fertilized egg. In this method, it involves a direct microinjection of a chosen gene construct. It can be a single gene or a combination of genes from another member of the same species or from a different species in the pronucleus of fertilized ovum. It is one of the first methods that proved to be effective in mammals. The introduced DNA may lead to the over or under expression of certain genes or to the expression of genes entirely new to the animal species. The insertion of DNA is, however, a random process, and there is a high probability that the introduced gene will not insert itself into a site on the host DNA that will permit its expression. The manipulated fertilized ovum is transferred into the oviduct of a recipient female or the foster mother that has been induced to act as a recipient by mating with a vasectomized male. A major advantage of this method is its applicability to a wide variety of species. Next technology involved is cloning. The reproductive cloning technique is used to produce multiple copies of animals, including transgenic animals, genetically superior animals, and animals that produce other desired traits. In reproductive cloning, the newly created embryo is placed back into the uterine environment where it is implanted to develop. To better understand this method, the reproductive cloning begins with somatic cell nuclear transfer. In somatic cell nuclear transfer, the scientists remove the nucleus from an egg cell and replace it with a nucleus from donor adult somatic cell, which is in any cell in the body except for an oocyte or a sperm. For reproductive cloning, the embryo is implanted into a uterus of a surrogate female where it can develop into a live being. Lastly, in addition to the use of transgenics and cloning, scientists can use the gene knockout technology to inactivate or knock out a specific gene. This technology creates a possible source of replacement organs for humans. The process of transplanting cells, tissues, or organs from one species to another is referred to as a xenotransplantation. Currently, the pig is the major animal being considered as a viable organ donor to humans. Unfortunately, pig cells and human cells are not immunologically compatible. Pigs, like almost all mammals, have markers on their cells that enable the human immune system to recognize them as a foreign and reject them. Genetic engineering is used to knock out the pig gene responsible for the protein that forms the marker to the pig cells.
History behind animal biotechnology. The traditional breeding techniques of animal biotechnology date back to 5000 BCE, which includes the technique of crossing diverse strains of animals to produce greater genetic variety. This process is known as hybridizing. Based on the crossing diverse strains of animals, its offsprings are bred selectively, which will produce the greatest quantity of desirable traits. As an example, during this era, the female horses are bred with the male donkeys to produce mules, and conversely, male horses are bred with female donkeys to produce hinnies. These types of animals are kept and trained by humans to perform tasks in the field for the past 3,000 years. Meanwhile, the modern era began in 1953 when the double helix model of DNA was presented by Watson and Crick and followed by Arbor's discovery of restriction enzymes in bacteria during 1960s. In this discovery, the enzymes cut the DNA strands of any organisms at specific points. During the modern era, a specific gene from one bacterium is removed by the American geneticist Stanley Cohen and American biochemist Herbert Boyer. The removed bacterium is inserted into another using restriction enzyme, which traces the beginning of genetic engineering. In year 1977, genes obtained from the other organisms were transferred to bacteria, which resulted to the first transmission of human gene.
To better understand the operation of animal biotechnology, we will be discussing some applications and issues of it. Here are what we will briefly discuss for its application: the protein property benefits of transgenic cloned animals, gene modification for human transplant, benefits of genetically modified products, and the possibility of transplantation of animal organs into humans.
Here are the applications of animal biotechnology. First is the therapeutic protein production in milk and blood of transgenic cloned animals. Over 20 years ago, the therapeutic protein production of transgenic mammals started from the microinjections of genetically engineered structures to the pronucleus of a mouse zygote. The production of transgenic cloned animals contributes to the development of livestock and production of recombinant proteins. The transgenic cloned animals are known for its ability to produce monoclonal antibodies. In year 2006, the European Medicines Evaluation Agency authorized the antithrombin, which is the first recombinant protein that is derived from the milk of transgenic goats. This type of protein is approved by the United States FDA as a drug category that prevents blood clotting to those patients with hereditary antithrombin deficiency. Another application is the use of animal organs in humans. Based on the studies of scientists from Massachusetts, utilizing CRISPR-Cas9 gene editing system can activate 25 viruses in the genome of pig, which strengthens the research on finding a possible source of alternative human organs. Xenotransplantation is a process of transplanting the cells, organs, or tissues from one species to another. Study shows that the organ systems of pig is the most visible organ donor to humans. However, human and pig cells are not immunologically compatible.
Another application of animal biotechnology is the gene modification of organisms to produce products and services like food, vaccine, or virus resistance in animal livestock. It was previously discussed how there's a therapeutic effect from the protein obtained from the milk and blood of transgenic cloned animals. Moreover, this technology called recombinant DNA had made its way as another breakthrough in biotechnology and healthcare. rDNA eliminates the use of live viruses. Instead, the DNA of the disease agent is disintegrated to its basic components to modify and inactivate it. It is then recombined and reinserted into the organism to produce a vaccine that is capable of inducing strong immunological protection or response. An example of this is the vaccine against hepatitis B and human papillomavirus or HPV. Animal biotechnology has also made it possible to produce animals more safely with unique properties for dairy and meat with enhanced nutrition. It is done through a process called transgenesis under rDNA technology. This practice has been performed and published in 2006, where transgenic cloned pigs were modified to possess an enriched level of omega-3 fatty acids. Omega-3 are especially beneficial to the health and well-being of humans. The emergence of animal biotechnology in animal livestock has reduced the adverse effect of the industry. There have been an increase in the animal agriculture efficiency and productivity by reducing the amount of land and water resources used. There would also be slower competition for human food. Animal biotechnology surely offers progress in different industries. Of course, not all technologies are 100% effective. Hence, some issues will be encountered with it, like compromised immune system, high rate of fetal death, and issues on health, environment, and animal welfare. Let's discuss the first issue. The immune system is compromised. The cells of animals and humans are immunologically incompatible. This suggests that when a foreign cell, tissue, or organ is introduced to the human body, the immunological response would be to reject it, which could be fatal. Moreover, animal cells transferred to humans could make their bodies susceptible to viruses known to the animal. The second issue is the high rate of fetal death. Reproduction of animals through cloning offers healthier offspring and more exact replication of the parent. However, present methods are not very efficient, especially in the case of cloning by nuclear transfer. Therefore, there is a high rate of embryonic, fetal, perinatal, and neonatal deaths among offspring. In any case where the offspring lives, some abnormalities may emerge. The third one are the unknown potential health effects to humans from food products created by transgenic or cloned animals, the potential effects on the environment, and the effects on the animal welfare. Aqua Advantage salmon is the first transgenic animal that has been approved by the US FDA for human consumption. It was genetically modified to grow more quickly. However, concerns arise like its allergenicity and unexpected changes in the composition of edible tissues. Animal biotechnology offers great advances, especially in securing the traits of endangered species and replicating their unique characteristics. However, this may be harmful to the environment in a way that biodiversity will be disturbed. There may be increase in the competition for limited food and resources in the environment, or changes in other biotic factors utilized. Lastly, animal welfare is a concern for animal biotechnology. Animals subjected into transgenesis undergo pain and distress, which causes behavioral abnormality and health problems. An example of this is isolation of pigs in xenotransplantation to minimize transmission of disease. This practice may result to abnormal behavioral development. To conclude this discussion, let's tackle on the status of animal biotechnology in the Philippines. In a report issued on 2017 by USDA Foreign Agricultural Service, it was stated that the Philippines was the first Asian country to allow planting of genetically engineered crop, the Bacillus thuringiensis corn. However, based on their outlook, no GA animals or clones are expected within the next 5 years. There's no regulatory framework or legislation for the development of animal biotechnology. However, recently on August 24 to 26, 2020, a webinar on training workshop on biotech animals for Philippine regulators was organized by CIRCA, USDA, ISA, and NCPB. It was to discuss and encourage the implementation of animal biotechnology in the country. The main part of the webinar was creating guidelines and legislation according to science-based evidence.